Imagine you are trying to read every medical textbook in the world simultaneously to find a cure for a disease.
Right, which is quite the undertaking.
Exactly. But there's a catch. Every single time you open a book, it costs you $1 million. And it takes a massive supercomputer a full week just to turn the page.
Yeah, and as wild as that sounds, that is essentially what the pharmaceutical industry is doing right now.
I mean, when they use massive artificial intelligence models to discover new drugs.
It is a massive bottleneck. We spent, the last few years relying on a totally brute force approach.
Right.
The philosophy was basically simple. Just buy bigger servers, feed the models more data, and hope the AI eventually stumbles onto the right molecular structure by sheer statistical volume.
But the stack of research we are diving into today shows that brute force approach is hitting a brick wall. So welcome to the deep dive.
Glad to be here.
Today, we are looking at the future of what the literature is calling pharmaceutical superintelligence. Now, if you follow biotech at all, you know there's already a very long list of flashy AI tools out there designing drugs.
Oh, absolutely. Hundreds of them.
But the real question for you, the listener, is, well, how do we make these tools actually affordable and fast enough to use in the real world?
Because if we don't, the technology just stalls out. I mean, we're witnessing a fundamental shift in computational biomedicine right now. The field is moving away from that brute force scaling and moving toward architectural refinement and data-centric precision. It's about fundamentally rewiring how the AI learns.
And this matters for you because this isn't just about saving some tech giant a few $1 million on their cloud computing bill.
No, not at all.
Reducing an AI's learning and thinking time is quite literally the difference between a life-saving drug taking 10 years to develop versus mere months.
Exactly. It's life or death in some cases.
Yeah. So the mission of our deep dive today is to explore how 3 specific architectural upgrades can be applied to almost all current AI biotech tools to solve this. We're looking at GraphMERT, attention residuals, and Polymath Learning.
Right.
Okay, let's unpack this. If we want to understand how these upgrades change the game, we need to know what they actually do. So let's start with GraphMERT.
Sure. GraphMERT acts as what researchers call a neurosymbolic anchor.
Hold on, I need to stop you right there. Large language models, LLMs, they're famous for being huge.
Right.
GPT, Claude, they all boast about having hundreds of billions of parameters. The conventional wisdom is just the bigger the model, the smarter the AI. So how does making something smaller actually make it better at complex medical science?
Well, it's actually a bit more complicated than just size. It is really about specialization versus generalization.
Okay.
A general LLM with, say, 32 billion parameters is fantastic at writing a polite email or generating a poem. But because it relies on probabilistic guessing, just predicting the next most likely word, it hallucinates facts.
Right, which you definitely don't want when you're making a cancer drug.
Exactly. GraphMERT, on the other hand, is an incredibly tiny 80 million parameter encoder-only model.
Wow, 80 million, that is tiny.
It's minuscule compared to the big ones. But it outperforms those massive models because it doesn't just guess words, it actually distills text into a leafy chain graph.
A leafy chain graph? Okay, I'm going to need a translation for that.
Think of a tree structure. The root nodes of this tree are the syntactic text tokens, like the actual words on a page of a medical journal.
Right.
But the leaves attached to those branches are semantic knowledge graph triples. These are verifiable, hard scientific facts.
Okay, so GraphMERT merges them.
Exactly. It binds the loose, flexible language of a medical paper directly to concrete, rigid biological concepts. It forces the AI to learn semantic units as complete entities, not just, fragmented word guesses.
So it's basically pinning words to hard science, which gives us some staggering numbers in the source material.
Oh, the numbers are incredible.
According to the research, GraphMERT hits a FACT score, which measures factuality of 69.8%. It also hits a validity score for ontology of 68.8%.
Yeah.
Meanwhile, the massive 32 billion parameter baseline LLMs are stuck down at 40% factuality. So we get a smaller model that is almost twice as reliable.
That's the first major shift.
Yeah.
But once you have accurate facts, the AI still has to process them through incredibly deep neural networks.
Yeah.
And that introduces a completely new problem, which brings us to attention residuals, or ATNRs.
Because medical data isn't just a single sentence. It's complex, it's layered data. The sources mention the vanishing gradient problem.
Hmm.
If I'm understanding this correctly, does the AI just sort of lose the plot when it tries to process too much deep information?
That is essentially what happens, yeah.
Yeah.
In very deep neural networks, information from the early layers gets diluted or lost by the time the system reaches its final output layer. Standard networks try to fix this with fixed additive residual connections. It's like trying to build a complex skyscraper, but the builders on the 50th floor have a blurry, faded copy of the original blueprint from the ground floor.
Oh, I see. It's an assembly line where the workers at the very end can't even remember what the original product was supposed to be.
Exactly, it degrades. But ADRS replaces those fixed, rigid connections with learned softmax attention.
Meaning the workers on the 50th floor can dynamically call the ground floor and pull the exact piece of the blueprint they need.
You're exactly on the right track. It allows each layer of a deep network to selectively retrieve relevant information from earlier stages.
Oh, wow.
Yeah, it dynamically weighs what is important at that exact moment in the computational process. And the efficiency gain is just remarkable.
The research shows an Attention 92 model can beat a standard ResNet 200 model using 32% fewer parameters, right?
Yes. So it's doing better, deeper work with a third less brain power.
Okay, what about the third pillar, Polymath Learning? Let me set up the problem here.
Go for it.
If we are looking for a cure for an ultra-rare disease and we just don't have enough data on it, how can an AI possibly learn to cure it? I mean, machine learning usually requires mountains of data.
And that is the core problem of extreme data efficiency. Polymath learning approaches this through what they call one-shot reinforcement learning.
One shot.
Yeah. Instead of needing thousands of data samples to learn a new domain, the AI uses a single, strategically engineered metasample.
A metasample. See, initially I thought of this as just a super textbook, but that doesn't really explain how it works. Let me try this. It's more like a decathlon.
Right, Cathlon?
Yeah, instead of training a runner, a swimmer, and a cyclist separately, you train one athlete in a synthesized, extreme environment where they have to run, swim, and bike simultaneously to escape a maze.
That is a much better way to conceptualize it, actually. The metasample combines mathematical skills like algebra and calculus with multidisciplinary contexts like physics, chemistry, and biology,
All at the same time.
all in one complex training scenario. By forcing the AI through that decathlon, it acts as a reasoning synthesizer. It learns the underlying logic of how to reason across different sciences rather than just learning how to memorize one specific task.
What's really fascinating here is how these three tools, GraphMERT, @Rees, and Polymath Learning, collectively change the foundation of how the machine learns.
Yeah, they do.
It's shifting AI from statistical guessing to actual verifiable reasoning. So let's take these upgrades and apply them to the tools that are actually mapping and building drugs right now in the real world.
Okay, let's look at the target discovery phase with a framework called AllSites.
Right. AllSites is a sequence-based tool, and it's designed to identify binding sites across all different types of drug modalities.
Exactly. And traditionally, if you want an AI to find a binding site on a new, rare protein, you need a massive, manually annotated dataset for that specific niche target.
And paying scientists to sit there and manually label 10,000 structural images of a rare protein is incredibly expensive and slow. It's highly prohibitive. But AllSites aims for structure-free efficiency. If we integrate polymath learning into it, the model can use what the researchers call polymath primes.
Polymath primes.
By using these synthesized multidisciplinary primes, all its sites can actually synthesize reasoning strategies for rare or out-of-distribution proteins without needing that mountain of historical data.
So it can essentially figure out where a drug should bind based on first principles, rather than just matching patterns from 10,000 past examples.
Precisely. That entirely removes the need for those expensive, manually annotated data sets. It dramatically reduces both the learning time and the operational cost.
And we see a different kind of optimization in the generation phase with a tool called Proteina Complexa.
Right.
This is a generative model for designing protein complexes, and it already outperforms older hallucination methods under normalized compute budgets. But Proteina Complexa relies on inference time optimization.
Also known as test time compute. Let me make sure I have this straight. Inference time means the AI has to think for a long time while it is actively being deployed, right?
Yes.
It's not just pre-trained, it's actively calculating its generation steps live.
Correct. And that real-time thinking is computationally very expensive. This is exactly where attention residuals step in.
Ah, Atners again?
Yeah, by injecting at-risk into Proteina complexa, you allow each layer of the model to selectively retrieve relevant structural information from earlier stages of its own live generation process.
Wait, so it doesn't have to keep recalculating what it already figured out 10 steps ago?
Exactly. It remembers the critical structural data. This drastically reduces the number of computational iterations needed to achieve in silico success for complex multimeric targets.
Wow.
You are effectively slashing the thinking time during deployment, which saves massive amounts of compute budget.
Here's where it gets really interesting. Because if we've optimized how the AI finds targets with allocytes and how it builds structures with Proteina complexa, the next step is simulating how those structures behave in the physical world.
Right, the simulation phase.
And we have to do that without burning millions of dollars in supercomputer time. Let's look at the unified platforms like ISODDE, which is Isomorphic Labs' drug design engine, and O-Design, an all-atom willed model.
Right. ISODE handles incredibly complex tasks like blind pocket ranking. Currently, they use high-parameter LLMs to read literature and rank these molecular pockets.
Okay.
But if they replace those massive LLMs with GraphMERT's compact 80 million parameter architecture, the operational costs drop by orders of magnitude, and their factuality jumps from that baseline 40% up to nearly 70%.
That is a massive jump. And for O-Design, which is designing all-atom biomolecular interactions, the bottleneck is the diffusion sampling steps. It takes a ton of computing power to reach atomic accuracy.
It does.
But the sources note that by using attention residuals, O-Design gets more stable gradients. In conversational terms, a stable gradient means the AI's learning process isn't spiking erratically, right? It has a uniform distribution of information across its depth.
Which means fewer sampling iterations are required.
So, faster thinking time.
Exactly. And if you add Polymath Learning to O-Design, it can suddenly work with entirely new modalities, like protein-binding RNA aptamers, with almost zero data construction costs.
Just because of that one-shot multidisciplinary reasoning.
Yes.
Okay, I have to push back here for a second. We're talking about molecular dynamics, how complex proteins actually fold and move over time in a fluid.
Yeah, the heavy stuff.
That is hardcore physics. Simulating that takes physical supercomputers months to map just a few microseconds of movement atom by atom. The AI must be cutting corners. What is it skipping?
Well, it is skipping a lot of the intermediary microcalculations, and it's doing it with tools like Deep Jump. Deep Jump uses a technique called Euclidean equivariant flow matching to predict long time-scale protein dynamics.
Whoa, hold on. Euclidean equivariant flow matching. You're going to have to explain that to me like I don't have a PhD in quantum physics.
Fair enough. Imagine trying to predict how a piece of crumpled paper unfolds in the wind. A traditional physics simulation calculates the air pressure on every single millimeter of the paper at every millisecond.
Which takes forever.
Right. Flow matching, broadly speaking, is an AI technique that learns the overall path from the crumpled state to the flat state.
Okay.
The Euclidean equivariant part just means the AI understands the 3D shape of the molecule, no matter how it rotates or moves in space. It maps the overall flow of the folding process without calculating every single atom's micromovement.
Which gives it that 1000x acceleration over traditional physics models the research talks about. But there's a catch, right? If it skips steps, it must lose accuracy somewhere.
It does. It actually struggles with accuracy on fast-folding proteins. It loses track of the fine structural dependencies.
I see.
But this is exactly where attention residuals come back in. If you integrate at NERIS, Deep Jump can utilize adaptive feature weighting. It can selectively pay attention to those crucial structural anchor points across multiple timescales, maintaining its accuracy while keeping that massive speed boost.
That makes a lot of sense. And there's another molecular dynamics tool mentioned in the stack, BioEmu. BioEmu generates conformational ensembles for kinases, which are crucial proteins in cancer research.
Yes, very important.
But the sources say it struggles with population shifts caused by disease mutations, like the BRAF V600E mutation.
Yeah, because standard models just guess based on latent hidden representations. But if you give BioEmu GraphMERT, you give it a structured layer of verifiable biological axioms extracted directly from the scientific literature.
So you are locking the AI's predictions to empirical evidence.
Exactly. The AI physically isn't allowed to predict a biological movement that breaks the known rules of physics or biology documented in those semantic graphs.
Okay, so we have our computationally efficient simulated drug. It binds, it folds, it respects physics in the simulation.
Right.
Now we have to make sure it actually kills bacteria, is safe for the human heart, and can actually be tested in a real clinical trial. Let's talk about the patient side, starting with antibiotics and toxins.
And this brings up one of the biggest roadblocks in medical AI, the black box
It's
problem.
right. I want a receipt. You wouldn't swallow a pill if your doctor handed it to you and said, honestly, I have no idea how this works, but my laptop said you should take it.
No regulatory body would approve that either.
Yeah.
We need to know the mechanism of action, the MOA. Tools like DiffDoc and ApexGo are great at designing antimicrobial peptides, but they have to explain why they work.
And that is exactly what GraphMERT provides, right?
Yes, it gives these tools verifiable MOA reasoning. It provides sentence-level provenance.
Wait, so basically it highlights the exact sentence in a published medical journal that proves why its predicted target is correct. It shows its math.
Precisely. And a similar upgrade applies to predicting cardiotoxicity. ADMeet AI is a tool that predicts drug-induced heart risks. Currently, it uses a standard XGB classifier.
Again, let's translate that. An XGB classifier is basically just an older, rigid categorizer, right? Like a highly advanced sorting hat.
That's a good way to look at it.
Yeah.
It sorts data well, but it limits the tool's ability to generalize to entirely new, unseen chemical structures.
Right.
But if ADMeet AI swaps that rigid classifier for a residual attention transformer, it can capture higher-order, complex interactions. It can trace exactly which features, like the inhibition of a specific enzyme, say CYP2D6, are causing the heart risks, without that crucial detail getting lost deep in the network.
If we pull back and look at the bigger picture, clinical trials cost billions of dollars, and they are a massive bottleneck. How does this tech help there?
We look at tools like AutoTrial and TrialGPT. AutoTrial designs the eligibility criteria for clinical trials, who gets in, who doesn't. But right now, it relies on general LLMs, which means it can hallucinate criteria. GraphMERT fixes this by grounding the trial criteria in a knowledge graph built from 70,000 precedent trials. No more hallucinations.
And TrialGPT, which matches patients to those trials, has to read incredibly long patient electronic health records, or EHRs. I mean, these things can be hundreds of pages long.
Which is the perfect use case for attention residuals. AtNeuris allows the model to process those massive longitudinal clinical notes without suffering from information dilution. It won't forget a crucial lab result from page 1 by the time it reaches page 50.
And what if it's a rare orphan disease where there basically are no patient records?
Then you use a tool called PyTrial, equipped with Polymath Learning. It uses that one-shot decathlon reasoning to simulate highly realistic patient records from almost zero starting data, allowing the try to actually proceed.
It even impacts the visual side, too. During these trials, you have MRI and CT scans. The sources note that deep neural networks used for analyzing these scans suffer from pre-norm dilution. I'm assuming that means as the AI analyzes the pixels deeper and deeper, it loses the sharp contrast of the original image.
That's exactly it. It loses the fine anatomical edges of tumors in its deeper layers. But AtNeuris allows visual tools like SwineNet to preserve those local spatial features and nodule boundaries.
Wow.
It allows the AI to keep its focus sharp no matter how deep the analysis goes.
So we've walked through the entire pipeline, finding targets, simulating physics, passing toxicology, and designing the clinical trial. But all of this still involves human supervision.
Surely.
The final leap in the sources is what happens when the AI basically runs the lab itself. Autonomous agents.
And we are already seeing the vanguard of this with systems like Superintelligence and Dynamate.
Yeah.
Dynamate is a multi-agent framework that runs its own self-correcting molecular dynamics simulations. But currently, it hits a hard limit. After about 35 iterations, it gets lost in the weeds and loses detail in multi-step actions.
But Polymath Learning fixes that. A single polymath sample combining coding logic with physics allows Dynamate to self-navigate software errors and parameter issues with far fewer trials. It learns how to troubleshoot its own mistakes.
Exactly. And for chemical process engineering, CPRO agents have to translate static, human-drawn engineering sketches into perfectly connected semantic graphs to design chemical plants.
Which sounds incredibly complicated.
It is highly complex topological work. GraphMERT's architecture ensures that the connection precision remains robust, capturing the implicit engineering rules without getting confused by the visual layout.
But all of this leads to the ultimate sci-fi concept from our stack of research today, the Darwin-Gödel machine, or DGM.
This brings everything full circle. A Darwin-Gödel machine is a theoretical self-improving agent that autonomously rewrites its own source code to improve its capabilities. It references Gödel's incompleteness theorems, the idea of a system stepping outside its own established rules to observe and improve itself.
And when you combine a self-rewriting DGM with polymath learning, The AI doesn't just learn from the meta samples we give it, it starts generating its own synthetic polymath primes. It
Yes.
creates highly efficient, perfectly engineered training scenarios to teach itself the next necessary leap in reasoning.
It bridges neural learning with structured symbolic representation. It achieves a true neurosymbolic self-evolution.
So what does this all mean? The era of just blindly throwing compute power at drug discovery, making models bigger and bigger, hoping they stumble on a cure, is ending.
It really is.
By using GraphMERT for factual grounding, attention residuals for deep, stable focus, and polymath learning for one-shot multidisciplinary reasoning, we are seeing a revolution. Tools like all-sites and Proteina Complexa are proving right now that we can drastically cut both the financial cost and the sheer thinking time of drug discovery across the board.
I would encourage you to really consider the magnitude of this shift. AI is no longer just memorizing data or finding statistical correlations. It is learning the fundamental rules of biology, chemistry, and physics to synthesize entirely new cures from first principles.
And I'll leave you with this final thought to mull over. If tools like the Darwin-Gödel machine start engineering their own scientific axioms, generating their own synthetic polymath primes to solve biological problems, what happens when these systems discover a verifiable, perfect cure for a disease, but it's based on a newly synthesized law of biology that human scientists aren't actually smart enough to understand?
That's the real question.
If the machine turns the page of the textbook and the text is written in a science we haven't invented yet, do we still take the pill? Thank you so much for joining us on this deep dive. We'll catch you next time.